Today, I am taking up part 2 of photosynthesis. The topic includes photosystems, non-cyclic and cyclic photo, phosphorylation, and chemiosmotic hypothesis. Then, photosynthesis involves two steps. In the first step, the solar energy coming from the sun is trapped and stored in the form of bond energy in ATP and also in NADPH. That is the first part. This is also known as light reaction. It is also known as light reaction or light dependent reaction. The second part is using this bond energy in the synthesis of carbohydrates and other substances. This second step occurs even in the absence of the light. That's why it is known as dark reaction or light independent reaction. Coming to the first part, that is trapping of the solar light, there are photopigments. So to trap the sunlight, we require photopigments. In the previous class, I have told you there are 4 types of pigments: chlorophyll A, chlorophyll B, xanthophylls, carotenoids, etc. These pigments, they form light harvesting complexes. Light harvesting complexes. All these pigments are present in the membrane system of chloroplasts. The membrane system is thylakoid membranes. The light harvesting complexes, or LHC, are mainly 2 types: photosystem 1 or PS1 and photosystem 2 or PS2. The photosystem 1 is known as P700 because at the wavelength of 700 nanometers, the activity is highest in PS1. In PS2, the activity is highest at the wavelength of 680 nanometers. So these are simultaneously known as photosystem 1 or P700, photosystem 2 or P680. Then each photosystem has 2 important components in the photosystem. One is light harvesting complex, other one is reaction center or antennal complex. See here, I have drawn a figure here. This is the light harvesting complex. So when solar light or sunlight falls on this complex, that energy is harvested. This light harvesting complex consists of all the photopigments except chlorophyll A. Chlorophyll A alone is present in reaction center. The reaction center may be 680 or 700. Then all the energy collected by the light harvesting complex is supplied to chlorophyll A or reaction center. In the reaction center, what happens? The electrons get excited and they gain energy. Usually what happens is, when electrons gain energy, they reach to a higher level, then after some time they lose the energy and come back to the original level. But here what happens is, the excited electrons are removed from chlorophyll. They get separated. That is a very important point. So in this way, the excited electrons get separated from reaction center. Now, let us see where these photosystem 1 and 2 are present. Here I have drawn thylakoid membranes. This is one granum, this is another granum. This is the thylakoid membrane. Here, 2 grana are joined by stromal lamellae. The stromal lamellae contain only PS1. Contain only PS1. That is, this part is excited only when the wavelength is at 700 nanometers. Whereas these thylakoids, they have both PS1 and PS2. This is PS1, this is PS2. So, stromal lamel has only PS1 and grana have both PS1 and PS2. That we have to remember. One more point is, the stromal lamellae, NADP reductase enzyme is also absent. NADP reductase enzyme is also absent. In the absence of this enzyme, PS1 cannot synthesize NADPH. Cannot synthesize NADPH. So now, photophosphorylation. What is the meaning of photophosphorylation? Photo means light. Phosphorylation means synthesis of ATP from ADP. So synthesizing ATP in the presence of sunlight is known as photophosphorylation. Photophosphorylation is two types: non-cyclic and cyclic. First we will take up non-cyclic photophosphorylation. This is also known as jet scheme, based upon the movement of the electrons. The electrons move in the direction of, in the shape of the letter Z. That's why it is known as jet scheme. Here I have drawn one thylakoid membrane. You must listen very carefully. In the thylakoid membrane, both PS2 and PS1 are present. Remember, these PS1, the names PS1, PS2 are given as these pigments were discovered, but not based on the role of their participation. Actually, in photophosphorylation, non-cyclic photophosphorylation, PS2, the role of PS2 comes first, then the role of PS1 comes next. This is not to get confused. So, this is PS2, where the activity is highest or at peak when the wavelength of light is at 680 nanometers. So now, when sunlight falls on PS2, what happens? Electrons, they get excited and they are removed from PS2, from this light harvesting complex. These electrons are to be accepted. Who will accept these electrons? There is one electron transport chain present in the thylakoid membrane, but it will not accept directly these electrons. So, before the electrons are transferred to the electron transport chain, some other substance or enzyme has to accept these electrons. So, these initial electron acceptors is plastoquinone, an enzyme. So, the enzyme plastoquinone is the initial acceptor of electrons. This plastoquinone enzyme is present on the surface of the thylakoid membrane towards the stroma. On the surface of the thylakoid membrane towards the stroma. Actually, plastoquinone is hydrogen acceptor or proton acceptor. It is the proton acceptor, but it also accepts the electrons. So this plastoquinone, by accepting electrons coming from here and also hydrogen ions from stroma, it travels through the thylakoid membrane. While traveling through the thylakoid membrane, these electrons are transferred to electron transport chain and hydrogen ions are released into the thylakoid lumen. Thylakoid lumen. So in this way, plastoquinone transports both electrons and hydrogen ions. Then, as the electron passes through electron transport chain, the energy trapped in the electrons is used to convert ADP into ATP. ADP combines with one inorganic phosphate and produces ATP. ATP is a high energy source. In ATP, energy is stored in the form of bond energy, which is made readily available for various activities. So by the time electrons travel from here to here, they lose the excess energy they have gained when exposed to sunlight. Then, in photosystem 1, again the electrons, the system is exposed to sunlight and these electrons gain energy. These electrons then leave the photosystem 1 and enter the stroma. In the stroma, these electrons along with hydrogen ions combine with NADP and they convert it into NADPH. In the presence of the enzyme NADP reductase. So with the help of the enzyme NADP reductase, NADP is reduced to NADPH. So in this way, during non-cyclic photophosphorylation, ATP and NADPH are synthesized. So in this way, non-cyclic photophosphorylation occurs. See here, the electrons are traveling from PS2 to plastoquinone present here. Then from plastoquinone through ETS, electrons travel to PS1. Then from PS1 they travel like this and enter the stroma. This movement of electrons resembles the letter Z. That's why it's known as Z scheme. Then, as the PS1 get excited when exposed to sunlight, it continuously loses electrons. Then what happens? After some time, there may be no electrons to get excited. To avoid this danger, photolysis of water takes place in the thylakoid lumen. Photolysis means splitting of water by using sunlight. So, sunlight is acting on both PS2 and also on water. HV means sunlight. The sunlight dissociates water molecules into electrons, hydrogen ions, and oxygen. So in this way, dissociation of water produces electrons, hydrogen ions, and oxygen. This molecular oxygen is released into the atmosphere. So in this way, in the atmosphere, oxygen gets accumulated and that oxygen is used by various animals and plants for the purpose of respiration. Then these electrons, they enter the PS2 and continue to get excited and move in this fashion. So, the source of electron supply to PS2 is water. So, water is the source of electrons to this one. Then what is the source of electrons for PS1? The source of electrons for PS1 is PS2. So the electrons coming from here, they enter PS1. This difference you must remember. So the source of electrons for PS2 is water. The source of electrons for PS1 is PS2. Then now let us see chemiosmotic hypothesis. Chemiosmotic hypothesis. This chemiosmotic hypothesis resembles osmosis. In the case of osmosis, what happens? Water moves across a semi-permeable membrane from higher water potential to lower water potential. Here also, the hydrogen ions or protons, they move from higher potential to lower potential. It means hydrogen ion gradient is established across the thylakoid membrane. This is thylakoid membrane. On one side there is stroma, on the other side there is thylakoid lumen. The hydrogen ions or protons are built up in the thylakoid lumen. That means they are in high concentration here and low concentration here. And how this gradient is brought about. Now let us see how it is brought about. When water splits during photolysis, what happens? I have told you already. Hydrogen ions are released and they remain in the thylakoid lumen. This is one way. Second thing, I have told you about plastoquinone. It is mainly meant for hydrogen ion transport. Along with hydrogen ion, it also transports electrons. So, for every 2 electrons it transports, it brings 2 hydrogen ions from stromal cavity into the thylakoid lumen. This is the second way. So, one way here, hydrogen ions are produced from splitting of water, and hydrogen ions are brought from stroma into the thylakoid lumen by the hydrogen carrier, plastoquinone. Then, in this way, hydrogen ions build up here. In the stroma, hydrogen ions are removed. Thereby, the concentration comes down. How it is removed? You see here. During the conversion of NADP into NADPH, hydrogen ions present in the stroma are used up. So, because of the use of hydrogen ions in the synthesis of NADPH, stroma loses some. Then some of the hydrogen ions are brought here, from here. So, by transporting hydrogen ions from stromal cavity into the thylakoid lumen, and also by incorporating hydrogen ions in the NADP reduction, the hydrogen ion concentration or proton concentration decreases in stroma. In this way, a gradient is established. Then, we know that the protons move from higher gradient to lower gradient. But there is a procedure for this. It just can't diffuse like this. For the movement of hydrogen ions or protons, one enzyme is required. That enzyme is known as ATP synthetase. This enzyme is also present in the thylakoid membrane. ATP synthetase has 2 parts, or 2 particles, FO particle, F1 particle. FO particle forms the transmembrane channel. Across the membrane forms a channel. This is the transmembrane channel. F1 particle protrudes from the surface of the thylakoid membrane into the stroma. So from the surface of the thylakoid membrane, it protrudes into the stroma. The F1 particle has enzyme. This ATP synthetase enzyme, which converts ADP into ATP when the hydrogen ions pass through this enzyme. So when the hydrogen ions or protons reach this point, energy is released and that energy is converted into ATP. In this way, ATP is synthesized as the protons or hydrogen ions move from thylakoid lumen into the stromal cavity. This is known as the chemiosmotic hypothesis. Then, there are 4 requirements for chemiosmotic hypothesis, or for chemiosmotic process to take place. 4 requirements are to be there. They are a membrane, this is thylakoid membrane, a protein pump, this one, proton gradient, I have told you, protons are in higher concentration here and a lower concentration there, and the ATP enzyme, this is the ATP enzyme present here. So in this way, these 4 requirements are to be there for chemiosmosis to occur. Then, in non-cyclic photophosphorylation, both PS2 and PS1 are involved. Both the complexes are involved. Then, electrons lost from PS2 are replaced through photolysis of water. PS1 receives electrons from PS2. From here to here they move. Both ATP and NADPH are synthesized in the stroma here. ATP synthesis is used to pump hydrogen ions. So here, proton pump means this one, not this one, this one. Plastoquinone. This plastoquinone pumps hydrogen ions from here to here. So it is known as protein pump. So ATP synthesized here is used up in pumping protons from here to here. That means this energy is utilized in the process of transporting protons from the stromal cavity into the thylakoid lumen. Whereas the energy, this energy produced here is used up in the dark reactions. That part will take up after, in the next chapter. Then, cyclic photophosphorylation. In cyclic photophosphorylation, only PS1 is involved. This cyclic photophosphorylation takes place when the intensity is low, when the light intensity is low, that is more than 680 nanometers. As the wavelength increases, the intensity of light decreases. So, when the wavelength of the light is more than 680 nanometers, this cyclic photophosphorylation takes place. Then, it occurs only when there is no fixation of carbon dioxide, or when the carbon dioxide is not utilized in the synthesis of carbohydrates. See, till now we have not seen the role of carbon dioxide. We have seen the role of chlorophyll, we have seen the role of sunlight, we have seen the role of water, but we have not come across the role of carbon dioxide. The role of carbon dioxide comes in the dark reaction. So, when the intensity of light is low and when there is no carbon dioxide fixation, this cyclic photophosphorylation takes place. Here what happens is, when the sunlight falls on PS1, whose intensity is low, electrons get excited and they are released from the PS1. These excited electrons are accepted by plastoquinone, just like here. The plastoquinone hands over the electrons to electron transport chain. So the initial electron acceptor is plastoquinone, then the electrons are transferred to electron transport chain, and as the electrons pass through the electron transport chain in a stepwise fashion, the energy stored in the electrons is released, and that energy is used in synthesizing ATP from ADP. In this way, phosphorylation takes place or photophosphorylation takes place. And when the electrons come here, they lose all the energy, excess energy. Then they get again excited with sunlight, and again they travel like this. In this way, they repeatedly travel in this fashion, in a cyclical fashion. So this is known as cyclic photophosphorylation. Now, one important point is PS1 participates in both non-cyclic photophosphorylation and also in cyclic photophosphorylation. PS2 participates only in non-cyclic photophosphorylation. That difference you must remember. Then, absence of carbon dioxide fixation results in non-requirement of electrons as NADPH is not being oxidized. So here, this step is absent. NADPH is not converted into NADPH because of the absence of carbon dioxide fixation. So that is about cyclic photophosphorylation. Then, again they have given points. How the proton gradient is established across the thylakoid membrane. Already I have explained. Again I explain one more time because it's very important. The proton gradient is maintained across the thylakoid membrane in three ways. First way is due to release of hydrogen ions due to photolysis of water in the lumen of thylakoids. So here, this step. When the water dissociates in the presence of sunlight, these hydrogen ions are released. Due to pumping of hydrogen ions across the thylakoid membrane from stroma into the lumen of the gland. This step. So this plastoquinone pumps hydrogen ions from here to here. So it acts as a proton pump. It acts as a proton pump. In this way, hydrogen ions decrease here and they get accumulated here. The third point, use of hydrogen ions in the synthesis of NADPH in the stroma. Here, in this step. These hydrogen ions present in the stroma are used up in the synthesis of NADPH. In this way, the hydrogen ions are reduced here. So here, in stroma, the reduction of protons occurs due to the action of the proton pump and due to the incorporation of hydrogen ions in the synthesis of NADPH. Then you may get one doubt. After some time, all the protons present in the stroma are lost. But it never happens because on one side, protons are removed. On the other side, these protons are coming from thylakoid lumen into the stroma through this protein, ATP synthetase. So in this way, there will be no shortage here. That is about photosynthesis part 2. In photosynthesis part 1, I have mentioned that the laughing gas was discovered by Priestley, and it is N2O, not N2S as I mentioned earlier. Okay.